Hello all, here are your chapter notes for Chapter 5, Cell Division. Cell division enables a single-celled fertilized egg to grow into an organism with trillions of cells. Very important, somatic cells are the body cells that continue to undergo cell division even as an adult. These include blood cells, skin cells, tracts that line the digestive system, respiratory tracts, and they are produced every day in life. Apoptosis is programmed cell death. This decreases the number of cells. This occurs during development to remove unwanted tissue. You can think of the tail of a tadpole or the webbing between human fingers and toes. And it also plays an important role in preventing cancer. The cell cycle is the orderly sequence of stages that occur between the time a cell divides and the time the resulting daughter cells also divide. So these are stages of interphase. So the cell cycle includes interphase divided into 3 stages. G1 is the stage before DNA synthesis. The S phase is where DNA synthesis occurs. And the G2 stage is after DNA synthesis. Then it enters the mitotic stage, which is the division of the nucleus, and then finally cytokinesis, which is division of the cytoplasm and when one cell turns into 2 cells. So here's a diagram outlining the stages of interphase, and there's 3 different checkpoints. There's a G1 checkpoint, a G2 checkpoint, and an M phase checkpoint. You can see here we have a growth phase, DNA replication phase, and then preparation for divisions as we enter into mitosis. G1, the cell doubles its organelles and accumulates material for DNA synthesis. In the S phase, DNA replication occurs, and after DNA replication, each chromosome has gone from one DNA molecule called a chromatid to 2 DNA molecules called sister chromatids. And these sister chromatids are 100% identical. In G2, the cell synthesizes the proteins needed for cell division and mitosis. So here's an example of a chromosome, consisting of one chromatid. It goes under DNA synthesis or replication, and you have 2 identical sister chromatids. Mitosis is the division of the nucleus, and it follows interphase. The sister chromatids separate into daughter chromosomes, and cytokinesis is the division of cytoplasm following mitosis. And the result is 2 daughter cells that are identical to the mother cell. Here's a diagram of apoptosis, and the cell passes through a series of events that brings about cell destruction. And the result is the blobbing of the cell on the far right into cell fragments. And there are different enzymes that are responsible for apoptosis. Apoptosis can occur by both internal or external signals. So eukaryotic cells have a complex system for regulating the cell cycle. So it can be controlled by both internal signals and external signals. And there are 3 checkpoints that control the cell cycle. In G1, the cell determines is the DNA damaged. In G2, the cell determines is DNA replication complete. And the M phase checkpoint determines if chromosomes are going to be properly distributed. And these checkpoints are critical for preventing cancer development. And a damaged cell should not complete mitosis and instead go through apoptosis. So once again, here is a diagram of the different checkpoints in the cell cycle. We have our G1, G2, and M phase checkpoints. And mammalian cells can enter the cell cycle only when stimulated by an external factor, such as a growth factor. And an example of this, when blood platelets release a growth factor, skin fibroblasts finish the cell cycle. So there are two types of genes that control progression through the cell cycle. The first are the proto-oncogenes, and these include proteins that promote the cell cycle and prevent apoptosis. You can compare this to the gas pedal of a car or a green light. These can be mutated to become oncogenes or cancer-causing genes. The other genes are tumor suppressor genes, and these encode proteins that stop the cell cycle and promote apoptosis. You can compare this to brakes on a car that inhibit progression through the cell cycle. When a mutation causes the brakes not to work, then cancer can proceed. So here's an example of, proteins turning things on for the cell cycle and turning things off, such as the proto-oncogenes, growth factors, and the tumor suppressor genes. So effect of a growth factor here, we see a growth factor is on the outside of the cell. It binds to its receptor. The receptor activates the RAS protein, which is a cell signaling pathway. It enters into the nucleus and turns on genes for cell division. On the right is how cancers form. We have a mutated RAS protein that turns on the genes for cell division, but no growth factor has stimulated it. So carcinogenesis is the development of cancer, and it is a multi-stage process. And it involves disruption of normal cell division and behavior. And so generally mutation initiates cancer, and it could be where proto-oncogenes become oncogenes, and it could also be the suppression of tumor suppressor genes. So proto-oncogenes can mutate to form oncogenes, which code for a protein that overstimulates the cell cycle. And mutated tumor suppressor genes code for a protein that fails to inhibit the cell cycle. So in mitosis, it's important to maintain the chromosome number. And eukaryotic chromosomes are composed of chromatin, a combination of DNA and protein, and these proteins are called histones. They are dispersed and extended in a non-dividing cell, and DNA is available for gene expression. But for cell division, they become condensed and compact. And histones play a role in coiling the DNA very tightly. So each species has a characteristic chromosome number. So diploid means the cells have a pair of each type of chromosome. So human body cells have 46 chromosomes in 23 pairs. And both the sperm and the egg are haploid. And I have one of each type of chromosome. And so the human eggs or sperm have 23 or one member of each pair. Mitosis is a division of the nuclear contents in which the chromosome numbers stay constant. One 2N cell becomes two 2N cells. DNA replication occurs before mitosis, producing duplicated chromosomes. So here's a brief overview of mitosis. You can see that the chromosomes are duplicated into sister chromatids. They then separate to form two daughter cells. So after DNA replication occurs in interphase, and that's very important, each chromosome is composed of two sister chromatids held together by a centromere. And these sister chromatids are genetically identical. They have 100% identity in their nucleotides. And during mitosis, the centromere divides and each chromatid becomes a daughter chromosome. And mitosis is nuclear division that forms two daughter nuclei with the same number of chromosomes and the same kind of chromosomes. The spindle of microtubule fibers brings an orderly distribution of chromosomes to the daughter nuclei. In prophase, the nuclear membrane starts to fragment, the nucleolus begins to disappear, the centrosomes have duplicated and begin moving to opposite poles, and a chromatin condenses and chromosomes become visible. Prometaphase kinetochores appear on each side of the centromere. The kinetochore of each chromatid is attached to a kinetochore spindle fiber. These spindle fibers extend from the poles to the chromosomes, and the fibers pull chromosomes back and forth towards alternating poles to begin aligning the chromosomes. So here's an image of mitosis occurring in animal cells. You can see early prophase. The membrane is starting to break down. The chromosomes are starting to condense. Prophase, we have duplicated sister chromatids that are starting to move around and line up. And prometaphase are getting ready to line up on the metaphase plate. So metaphase, a spindle is fully formed and consists of poles, asters, and fibers. And the pole fibers overlap. The centromeres of the chromosomes are aligned at the metaphase plate. In anaphase, the centromeres divide and sister chromatids are moved to opposite poles by fibers. In telophase, spindles disappear, the nuclear membrane components reassemble around daughter chromosomes, and chromosomes become more diffuse again. The nucleolus appears in each daughter nucleus, and cytokinesis begins. So here are some images of starting with metaphase. The chromosomes align at the metaphase plate, and then they're getting ready to go through cytokinesis on the very far right, where one cell starts to divide into two daughter cells. All right, mitosis in plant cells. It prevents the growth and repair as in animal cells, and it goes through the same phases as animal cells, but it does not use centrioles or asters. So here are some phases of mitosis in plant cells. We see early prophase, then prophase, and prometaphase, very similar to animal cells. And we have metaphase, anaphase, and telophase, just like in animal cells, so very similar. So cytokinesis in animal and plant cells. So cytokinesis in animal cells, a cleavage furrow forms between the two daughter nuclei. A contractile ring of actin filaments constricts, deepening the furrow, and the process occurs until separation is complete. So here we see the formation of the cleavage furrow and a contractile ring and the separation of the two cells. So cytokinesis in plant cells requires the creation of a new cell wall between the daughter cells. And here we have a flattened small disc appears between daughter cells, and vesicles move to the disc and fuse. A cell plate then forms, and then we're getting ready to turn into two new plant daughter cells. So here's a cell plate and a cell membrane by an electron micrograph. So meiosis is very different than mitosis. It's reducing the chromosome number. And this only occurs in sexually reproducing organisms. It reduces the chromosome number in half and provides offspring with a different combination of traits from that of either parent. And this only occurs in the egg and the sperm in humans. So it begins with one diploid parental cell, requires two cell divisions, and ends up with 4 haploid daughter cells. One chromosome of each homologous pair inherited from one parent, the other inherited from the other parent. So in meiosis 1, homologs line up side by side in a process called synapsis. Pairs of homologous chromosomes align on the metaphase plate, and when the homologous pairs separate, each daughter cell receives one member of the pair. The cells are now haploid. So in meiosis 2 and fertilization, no replication of DNA occurs between meiosis 1 and meiosis 2. the centromeres divide and sister chromatids migrate to opposite poles and become individual chromosomes. Each one of the 4 daughter cells has the haploid chromosome number, and each chromosome is composed of 1 chromatid. So here's a diagram showing one diploid cell that in the end, going through all of meiosis, turns into 4 cells, and they are haploid in nature. So we went from 2N to 1N. All right, fertilization. So daughter cells of meiosis mature into gametes, so the sperm and the eggs, also called sex cells. And gametes fuse during a process called fertilization and restores the diploid number from 1N to 2N and creates a cell that will develop into a new individual. Remember, meiosis requires 2 nuclear divisions and results in 4 daughter nuclei, and each daughter nucleus has half of the chromosomes as the parent cell. So the first division goes through prophase, anaphase, metaphase, and telophase, and meiosis is important to ensure genetic variation. This occurs in 2 ways, by crossing over and independent assortment. So here's a diagram just outlining the first division of meiosis. This diagram is continued. So in prophase 1, the spindle appears, the nuclear envelope fragments, and the nucleolus disappears, very similar to prophase 1 in mitosis. Homologs pair up during synapsis, and this is where crossing over can occur, the exchange of genetic material between non-sister chromatids of homologs. After crossing over, the 2 chromatids of a chromosome are different. One has the original genetic material, and one has recombined genetic material. And here's a diagram showing the crossing over event, and you can see here that we have recombinant daughter chromosomes, so their genetic material is a little different. So in metaphase 1, homologous chromosome pairs line up at the metaphase plate, such that maternal or paternal member can be oriented towards either pole. And in anaphase 1 is when independent assortment occurs, and these homologs separate and are pulled to opposite poles by spindle fibers during anaphase 1. So independent assortment generates cells with different combinations of maternal and paternal chromosomes, the mommy and daddy chromosomes. So in humans with 23 pairs of chromosomes, the number of possible combinations is 2 to the 23rd power, or 8,388,608 possibilities. And this value does not include genetic recombination due to crossing over. So independent assortment during meiosis 1, there are 2 possible orientations of chromosome pairs at the metaphase plate shown here. Depending on the species, it may or not occur at the end of 1, as this is telophase 1. The nuclear envelopes reform, the nucleoli reappear. Cytokinesis may occur, producing 2 daughter cells, which are haploid, and daughter cells have 1 chromosome from each homologous pair. So the term interkinesis is the period of time between meiosis 1 and meiosis 2, and there's no DNA replication going on here. So in meiosis, we have a second division. So in prophase 1, cells have 1 chromosome from each homologous pair. A spindle appears, the nuclear envelope disassembles, and the nucleolus disappears. Each duplicated chromatid attaches to the spindle, and in metaphase 2, those chromosomes consisting of pairs of sister chromatids line up again at the metaphase plate. In anaphase 2, the sister chromatids separate and become daughter chromosomes. Telophase 2, the spindle disappears, the nuclear envelope reforms, and cytokinesis then occurs to produce 4 haploid daughter cells. So here is a diagram of the second division that occurs during meiosis. Diagram continues. The second division occurs. So then what is the importance of meiosis? So meiosis produces haploid cells from diploid cells. It allows for genetic variation and produces cells no longer identical to the parental cell, which is different than mitosis. And the genetic variation can occur in two ways: by crossing over between non-sister chromatids and the independent assortment of chromosomes during anaphase 1. So upon fertilization, combining of chromosomes from genetically different gametes helps ensure offspring are not identical to parents, and this genetic variability is the main advantage of sexual reproduction. And so this long-term genetic variation increases the survival of a species. If you compare meiosis with mitosis, DNA replication only occurs once prior to either meiosis or mitosis. Meiosis requires 2 nuclear divisions, mitosis requires only 1, and meiosis produces 4 daughter cells, and meiosis only produces 2. The 4 daughter cells from meiosis are haploid, and 2 from mitosis are diploid. And really important here, the daughter cells from meiosis are genetically variable, while those from mitosis are genetically identical. So here is a diagram that you all can look at on your own, looking at meiosis versus mitosis. This is figure 5.16 from your book. And you show how the cells progress through both meiosis and mitosis. So it's important to realize that meiosis only occurs at certain times in the life cycle of sexually reproducing organisms. And this is after the reproductive organism matured to produce gametes. And mitosis takes place almost continuously in all tissues as part of growth and repair. So you can go through this on your own. These are the comparisons of meiosis 1 to mitosis. And the next slide is a summary chart comparing the two, which I think is quite nice. There's a comparison of meiosis 2 with mitosis. During the human life cycle, both mitosis and meiosis are required. At fertilization, a sperm and an egg fuse. They're both haploid. The resulting zygote is diploid. The fetus divides by mitosis for growth and development. And after birth, mitosis allows continued growth and tissue repair. So meiosis in the testes of males is called spermatogenesis, and it produces sperm. And meiosis in the ovaries of females is called oogenesis, and this produces the eggs. So here is a diagram of the life cycle of humans. So spermatogenesis occurs in testes of human males. The process begins at puberty and continues out through life. Primary spermatocytes divide in meiosis 1 and form 2 secondary spermatocytes. Then these secondary spermatocytes divide in meiosis 2 to produce 4 spermatids. These spermatids then mature to form mature sperm or spermatozoa. So here's a diagram here of spermatogenesis. Oogenesis begins in the ovaries of female fetus. All primary oocytes are arrested in prophase 1, and it resumes at puberty. one primary oocyte continues the process of meiosis during each menstrual cycle. So it produces primary oocyte 2 and divides into meiosis 1 to produce one secondary oocyte and one polar body. If the secondary oocyte is fertilized, meiosis 2 will proceed. If the secondary oocyte is not fertilized, it disintegrates. Here's a diagram for oogenesis. The comparison of spermatogenesis and oogenesis. And this concludes our chapter.